Okay, תודה רבה. Thank you, Yoram. Good morning, everybody. My talk actually is concentrated on two topics. One is nanoscale crystallography. Second topic is on soft matter corresponding to nanodroplet and nanobubble research. And both were carried out using transmission in the scanning electron microscope. And I'll just say it in short, SEM. And maybe the very basic question that you may ask yourself, why to carry experiment in the SEM using transmitted electrons? Because SEM is basically configured for bulk materials, and therefore most of the information is obtained by reflected signal. And this is true, that most of our work is done on bulk materials. But in case that the sample is thin enough, you can also have transmitted electrons. And then what do you gain by that? First is the issue of resolution. You still get 1 nanometer resolution, but something around 1 nanometer, but could be improved over what we have in the reflected signal. And the exact value actually depends on the sample configuration and on the system itself, the microscope itself. What is not less important is the issue of contrast, because in amorphous materials, non-crystalline, we have a mass thickness contrast in the bright field or dark field images. If you look at this equation, which is simplified equation for dark field, you see contrast dependence on the energy of the beam and on the atomic number. So in case that we are working at low energies, like in the SEM relative to conventional TEM, we have actually better contrast. And it is particularly important for low atomic material like biological materials and soft materials. So this is quite known about transmitted information in the SEM. And I would like to talk about two additional topics, something beyond this conventional knowledge. So first of all, I would like to talk about liquids. How can we work with transmission in liquids? These are actually bubbles, and I'll tell the story nanoscale bubbles or 10 nanometer bubbles, and I'll tell later the story about this research and configuration. In addition, we can do some crystal information or obtain some crystal information at the nanoscale using transmitted electrons. And this is a configuration just to show you, not just to know the crystal structure, but orientation. And here we have a very powerful tool to guess orientation at the nanoscale. So this will be the two topics that I would like to talk about. And first one is on actually droplet and nanobubble research using wet scanning transmission electron microscope detector in the environmental scanning electron microscope. So this will be sort of an overview of in-situ formation of thin films, nanodroplets, and I would like to show the similarity with nanobubble research. And actually to put forward a very physical question about the long lifetime and delay coalescence of nanodroplets and nanobubbles. Second part will be on crystallography by transmission Kikuchi diffraction, and I would like to show applications from Tel Aviv University and with collaboration from the last 2 years, and some special effects, interesting effects about thickness variation and contrast inversion. So let's start with the first issue of the in-situ nanoscale droplet or bubble research. So this is actually picture taken with the STEM detector in the environmental scanning electron microscope. And what you actually see here, small bright spots, these are bubbles. These are actually magnetic bubbles. And we can see that these bubbles actually survive at length scale of imaging, let's say seconds, it can survive for minutes. And this is in a loud magnification showing that these actually bubbles are of microscale. And they actually have a shell, which is a magnetic shell, because they are used for ultrasound applications, biological applications. But actually bubbles which are without such shell can have actually lifetime which is similar. So, but theoretically, it is expected that micro-sized bubbles actually won't survive that length, that actually they have time life of microseconds, okay? And it actually very much limits the application for biology or material science. And if we look, the Laplace pressure goes as 1 over the radius, and if we look at submicron or nanoscale bubbles, so we understand that it becomes even more problematic. But experiment like this and others shows that the lifetime is much longer, and this is a really interesting physical question to try to understand how really it can be. So, there are actually, from the point of electron microscopy, there are actually 2 approaches to answer this question in order to follow the dynamic events happening in wet samples. And this is shown here on TEM configuration, on a transmission electron microscope configuration, but the same holds also for scanning electron microscope. We have the window technique, in which the sample, liquid sample, is inserted in between 2 E-beam transparent windows, so that the rest of the system is kept at high vacuum and only that area is kept at high pressure. And the second approach is the aperture limiting approach, in which the sample is sandwiched in between 2 apertures, above and below, and there is some pressure gradient that is maintained by differential pumping. And the system that I was using was a sort of aperture limiting technique. This is the environmental scanning electron microscope. That the system itself is actually split into few vacuum regions that are shown here by different colors. So if we look at the site of the sample and we enlarge it here, we see that there are certain apertures which are above the sample and actually provide this pressure gradient. So in case that the sample is thin enough, we can have a configuration very much like transmission configuration, so that we have a thin sample, we use a TEM grid, and finally we get information in bright field or dark field. So this is a TEM grid, and this is a sample, an example, that we have here a wire on which there is some condensation of vapor. So basically the images that we obtain in SEM or TEM are like 2-dimensional images. But there is more information in the image. There is information for the 3rd dimension, which is called mass thickness information. And this is what we actually want to derive out of the picture in order to know what is really happening to this droplet and what is their exact shape. So I will show a procedure that I was going through in order to try to answer this question, although there could be other procedures as well that could be taken. So I'll show a quantitative approach toward droplet and thin film shape characterization. First step was to form very thin liquid layers. And here you have a grid, and I put actually a liquid, this is a water droplet, on top. And you can see that with time, and it could be also with some help of pressure changes, we can perform a receding of the contact line as you see here. Finally, we can end up with a grid which looks almost dry, but is just almost. When we zoom on one of these quarters here, we see the shape of this particular grid, which is a polycarbon structure. And if you look very precisely, you can see some contrast here that is different from the other regions. These are actually very thin liquid films trapped inside. And to make the story short here, I would say that the thickness of these liquid films is around 10 nanometers. So if you compare it to the situation before putting the drop, you can see the difference here. And it means the trapping of liquid films still occurs, and it could be done in sizes of around 1 micron, at least at one dimension. So second stage was to perform condensation. This is actually a pressure temperature phase diagram of water. We can work, for example, at a constant temperature and elevate the pressure and perform condensation on the material. The third stage was actually to run Monte Carlo simulation for what we actually have. And the simulation shouldn't be on actually bulk material, which has solved software, but should be on thin films or special structures, and this should be written particularly for the specific sample and detector geometry. And by doing that, we can find out thickness, droplet shape. Let's see how it works practically. This is an example of the holy carbon grid. You see here trapped liquid films, and on it some condensation of liquid droplets. So on the film, on the grid itself, on the liquid film. And in this particular case, I added some polystyrene particles of 100 nanometer to form calibration. Now, if we take a line scan across the drop and the polystyrene particle, we can have here the normalized intensity after taking the calibration according to the polystyrene particle. So this blue curve is normalized intensity, actually experimental intensity, normalized. And from that, we can run a simulation to fit the curve for the drop, and we can find actually the thickness. And what we find out is actually that these are not spherical drops, but actually of spherical cup geometry, which means actually that the radius of curvature is much bigger, which can explain why these drops could be more stable. But this is only first step in the understanding. Second step is to run some dynamic work. And as you know, dynamic nucleation usually goes into competing mechanism of heat transfer. One is the filmwise nucleation, which is the heterogeneous growth, and the second is the dropwise nucleation, which is the heterogeneous growth. And this is homogeneous growth, this is heterogeneous growth by the dropwise nucleation, and this growth is actually preferable in terms of heat transfer because it's more efficient for heat removal. So we will see both in situ how they could be run in the configuration that I showed. So these are initial stages of filmwise condensation. We have here the film, the liquid films, of very thin liquid films. We can see that by elevating pressure, we can form condensation, which starts from outside in. You see this darkening of the area around, and it can continue under equilibrium even, and you have a thicker film and a wider film. Finally, this film can have result with some instability, and it can break into droplets. Second procedure is for single droplet condensation, and these are the really first steps or first seconds of the condensation on the film. This size here was software measured to be 10 nanometer. You see that in the initial state, the vapor is not very well organized, but after a few seconds, we have this circular arrangement, which is the more equilibrium arrangement that we actually are familiar with. So let's look at nanodroplet growth behavior. So this is taken from a whole video for growth behavior, and this period of time between 13 to 27 seconds are actually under equilibrium, obtained under equilibrium without changing pressure temperature in the system. And you can refer to two droplets that are attached actually on the film, but actually at the side of the grid. And we can follow the time dependent of the growth. And what we actually find here is actually the parameter of fit, which is around half, and or below, and it really fits to the growth, which comes from vapor coming to the perimeter of the drop. But what is not less interesting is actually the fact that we have two parameters. So it means there is something additional in the system that is not consistent, and this is actually the pinning effect. So now we can understand the stability is not by the curvature and the shape of the drop, but also by the pinning effect to certain sides in the actually configuration, the specific configuration. And in addition to this long lifetime, we have also long coalescence, and we can see here again. This was taken from a movie. This is equal 58 seconds. We see here 30 nanometer spacing. We see flat facet as should occur, but very long lifetime, and then some coalescence which occur within 1 second, but still the liquid is not very well organized. Things take time. Okay. And while this thing was published 2014, in the other area in the world, in Singapore actually, in the National University Singapore, they were performing very similar experiments, but in actually different setup. They actually used the window technique, and they put a drop, a very thin drop, you see here the scale, and inside they put some really nanometer gold particles. And what they actually measured is the movement of these gold particles, and according to time scales and distances, they actually concluded that the diffusion constant is very, very small, and they talked about very high viscosity, and finally they made a conclusion that there is also pinning to the substrate. So conclusion was very much similar. And another group actually was working at the same time on nanobubble characterization. Here they provided me the sample, which was a graphene sample in which there was oxygen bubble encapsulated, not the regular what we actually are familiar with, the hydrogen bubbles which are by radiologists, but here are the oxygen bubbles that were initiated by ammonium persulfate interaction in water. And here you see the SEM image. This is the surface. Actually, you see a pocket. And when we look at the transmission information, what we actually see are really bubbles of 10 nanometers. And they are quite stable at the time of the imaging. And if we look in another area, we see again the same features. You see this, how they evolve, how they make some coalescence. They can make bubbles of 50, 100 nanometers, but here are smaller ones, and this is quite stable. So we see that in non-saturated liquid environment, and we can have some stability in bubble formation. And in addition, this group was doing TEMEM measurements, and they found out, this is part of the thesis that was done, actually a coalescence lifetime of sub-seconds, and they also followed single bubbles that looks like spherical in a non-saturated environment, and what they saw, some stability over time scale of seconds. So in case that this issue is really correct, that these are free bubbles, so it's a really a big story here, and all this issue is still under continuous research. So here I showed some combined research of TEM, SEM, also resemblance between bubbles and droplets in the research. Now I would like to switch to the second topic, which is crystallographic using transmission Kikuchi diffraction. So usually, as I said, we are getting information from bulk materials using reflected signal, and this signal could be signal for backscattering as well. And actually, electron backscatter diffractions are specific electrons that contribute. And in this case, the information comes from the upper surface of the material. But in case that we tilt the sample 90 degrees to this direction, we can have transmission information in this off-axis configuration. And then we have Kikuchi information, which is called the TKD, transmission Kikuchi diffraction, from the bottom surface of the material. And what we gain by that, much better resolution. Why, actually? Because if you look at the two different configurations, you see much more scattering in the EBSD than in the TKD, and we can also see that the intensity, this is the intensity electron distribution, the intensity is much more sharp and defined for the TKD orientation. So it actually explains us why we have a much better resolution in this configuration, which actually is attributed to the fact that we have a thin film and we have a low tilt angle in orientation. So how really to specify the resolution? It is usually specified both for reflection and transmission over a crossing grain boundary. So this is one actually grain, and we have one solution. This is the Kikuchi pattern, one solution, and we have another grain with the other solution, but at the interface we have overlap. And this overlap is not well resolved by the algorithms, and it actually was dictated the resolution. But now algorithms are becoming better, and hopefully we can somehow overcome. But the situation, the moment is the following. If this is around 100 nanometer thick nickel cobalt film, we have here all the unresolved information in green. And if we thin it to half, we can have a better resolution. So now you may think that we can thin the sample more and more and more and then get better resolution. But it's not that way, because there is a minimum thickness for the whole generation of these Kikuchi patterns. And it is very well emphasized experimentally here, although it is really very well understood also theoretically. So this is about 100 nanometer diamond film where we have very nice Kikuchi pattern, and when we thin it down to 30 nanometer, we having this spot information, which is not very well resolved and actually interfere what is called the half transform for understanding or recognition of the line patterns in the Kikuchi. And very nice and interesting result here is about contrast inversion. We are very regular to this picture where we have a high intensity at the center, but when we move about and around, we may have also contrast inversion. And this contrast inversion is actually an effect on the outgoing beam. And it also occurs in reflection, and very interesting to show how it was simulated in order to understand what is happening. And this is simulation done by Amo Wickelman, very nice simulation, taking dynamic diffraction into account. And what he actually showed here is that the regular pattern that we obtain is actually superposition of type 2 and type 1 Bloch waves. The type 1, as you remember for solid state physics, is for electron going on the planes, and type 2 for electron going in between planes. And superposition, this is what gives us this contrast. But in case the type 1 is somehow absorbed, and in the, let's say, simulation, you can put just a meter which is well below the surface. But if it is somehow absorbed, this type 1, absorbed by, of course, interaction, inelastic interaction, then type 1 decays, and therefore you have this contrast inversion. So this whole story of diffraction is very, very interesting also from the physical point of view, and I would just want to conclude this part by showing some results that were obtained, recent results that were obtained or actually published already in the last 2 years with the help of many, many people around, and I'm going to mention. So this results corresponds actually to 3 types of preparation methods, because as you might understand from here, the preparation method is very crucial, and the thickness that we're working on very crucial for the success of the results. So we can work with as-grown materials. This could be sputtered or self-assembly materials. We can work with neutral or induced thickness gradient materials. Materials that were prepared by ion milling to have a thickness gradient. Or we can work with nano-sectioned materials by obviously SEM fib, those that have it, or by ultra microtom that was done in Tel Aviv University very, very successfully. So I'll show all the 3 types of material preparation and what was obtained for them. So the first is the as-grown. These are biominerals of actually calcite that were obtained by Kokolis organic macromolecules, and this was done by Asaf Gal from the Weizmann Institute, who brought the sample. Asaf actually had that information already about the TEM, from the TEM, about crystallography and AFM information. But what was missing here was orientation from the whole feature of the 1 micron size biomineral. And what he was just asking to know, what is the orientation really of this hexagonal structure? And what you see here is actually a pole figure of actually inverse pole figure showing orientation of the 001 direction, okay? And it seems quite uniform. What I want to emphasize here, this is the pole figure showing the orientation in plant, and what I want to emphasize here that the step size for identification was 70 nanometers actually. But we could reduce it actually to 10 nanometers. This is the appropriate length scale for this, doing this TKD, and this is the advantage of the TKD. But as I said, it is sufficient. It is actually very excellent, and that was where we stopped, actually. And second example is for samples that were produced in the lab of Noam Eliaz by the student Sigalit Ifergan, and she was preparing stainless steel samples, and she actually investigated the aging process on these samples and relative to hydrogen diffusion and corrosion and resistance. And and it was prepared by doing ion milling for thickness gradient. What we see here is actually in gray the mapping of reverses austenite in the Martensite matrix. So reversed austenite is shown here in gray. And you see that as the aging temperature increases, we have much more population of this phase, okay? And in addition, what is very important in the TKD, the information that you can get is the localized information where this phase sits. It's not just XRD that can tell you that it exists or increases, but the position where it is, okay? So this is a great benefit. What you see, the colors, it is for the Martensite phase, and actually the spot size here was 8 nanometers, which shows you really the power of this method, and therefore also this poster presented got the second prize in the Royal Microscopy Society 2 years ago. And so it's really very powerful, and I want just to remind that we were in Tel Aviv the first to implement this method in our lab, and then now many universities are taking same steps. And the third example is from the lab of Fernando Potolsky. He's a PhD student, Alon Kozlov, who's doing this excellent work of preparing these materials, growing them, these pillars of core shell silicon metallic silicide, and he was a heterojunction, and he was slicing them with a microtome, and we were taking the TKD information. So what you see here, and this is really nice, and pay attention because it's like a flower really. So you see the inner side, this is the silicon in blue, and you see the outer side, this is actually the silicide. And this is a band contrast image that you can obtain. And what you see here, 3 grains, 3 orientations, very powerful presentation to see it. And if we look in this polygon configuration and we look for the 100 direction, you see that if it is in the silicon perpendicular to the sample, the 100 for the nickel silicide is a bit deviating. So very easy way in order really to get orientation information, and not that easy in other type of methods. And very good resolution, really not atomic resolution, but nanometer resolution, which is very sufficient for many, many applications. So I would like to conclude this work, this part, actually presentation, and saying that we saw both bubble and droplet are actually characterization by correlating between the SEM and TEMEM results, and I try to show some correlation between behavior of nanodroplets and nanobubbles, and there are still unresolved issues concerning the lifetime and long lifetime of bubbles in many experiments. Just want to say a few words about crystallography. I believe that we are at a stage that TKD and SEM assists very much conventional TEM crystallography study, and there are very interesting issues about the thickness effect involving contrast inversion and dynamic simulation. I would like to thank all the people being involved in this work, to thank you for your attention, and to conclude by that.
Thank you very much. We're behind time, so maybe 1 or 2 quick questions.
Zahava, thank you for your talk. Can the method for the bubbling analysis can be used?
Can the method for?
The bubble.
Bubble, yeah.
Can be used for learning about self-assembly?
Yes, you see the critical limit here is a technical limit, because in the transmission we can have resolution of 1 nanometer, but in the wet we are a bit more limited about the instruments and very much dependent. In the instruments that we have in our lab, we have, I believe, not better than 10 nanometer. But I think that in principle it could be reduced, because this is not, this is just a technical limit concerning stability. So in case that you have self-assembly, there are 2 points that you need to take into consideration. One is the temperature of work that it occurs if we can reach this temperature, meaning that as we elevate the temperature, we need to elevate also the pressure working, you see. So this is the first thing. And second is the resolution that these actually particles actually reorganize. What is the critical dimension that you need? if it is 10, if it's 5, if it's 2. So. Okay.
More questions? So thank you very much.